Great.
Okay, we're now recording.
Okay. Cool. All right, so we can get started, and then if anyone is joining, hopefully they won't miss out on too much of our exciting introductions. But yeah, so Jamie and I are going to be talking about the experiment that we worked on together at BU, studying the effects of climate change on New England forests. And we'll get into a little bit about what that means, but I'll let Jamie introduce herself.
Hi, everyone. Some of you, I know it's great to see the familiar faces, but if I don't know you, my name is Jamie Harrison, and I completed my PhD last year. Actually, my year anniversary of my defense is coming up. I defended on May 1st of last year in the middle of the pandemic. And I did my PhD at Boston University. And we're going to talk a little bit about my research, and afterwards, if anyone has any questions, happy to talk about that. And right now in my current position, I am a postdoctoral researcher at Columbia University and at the Lamont Doherty Earth Observatory. And so what we're talking about right now, Meg and I are going to talk about like our field methods and how we got our hands dirty hiking through the forests of New England. And right now what I'm working on is focusing on atmospheric gases. So I'm sort of moving up in the canopy a little bit, and I'm working at Harvard Forest in Massachusetts, and then also Howland Forest in Maine, looking at the cycling of gases. So CO2, and then this also lesser known gas of carbonyl sulfide, which can kind of be used as a proxy for carbon dioxide cycling, which I'm also happy to talk about a little bit more, but that's sort of where I am now.
That's so exciting. And I'm Megan Blagden. I also went to BU for undergrad, which is where I met Jamie in our lovely lab. I studied Earth and Environmental Science, and I graduated last January. So somehow I managed to avoid taking classes in a pandemic at the last possible second, which I am very grateful for. But I do miss Boston. And ever since I graduated, well, last year in the fall, actually, I got in contact with David, the executive director of Speak for the Trees, because I had heard about Speak for the Trees in college. It was on my radar of good things happening in Boston, and started volunteering in the fall with some fundraising efforts. And now I'm interning through the Mass CEC to work on lots of exciting fundraising, lots of fun, lots of fun things. So we'll get into some intro climate change 101, and just sort of explain the context of how or why we study climate change in forests together. So as you all probably already know, the atmospheric concentration of CO2 has been steadily and rapidly rising for over the past 100 years. This is just a little snapshot of the recorded concentrations in Mauna Loa. And this is largely as a result of human activities such as factory and industrial emissions, our car emissions, and deforestation, all of which increase CO2 into the atmosphere.
And this is also important in how it relates to incoming radiation from the sun. So incoming shortwave radiation from the sun, as you can see on this figure here, is absorbed by clouds, land, and by water. And some of that is also reflected as long wave radiation, which you can sort of see at the bottom of this figure here. And next slide, Meg. So some of that long wave radiation that is reflected out can sort of leave our atmosphere. That's what we like to see. But then some of that can be absorbed by greenhouse gases that would otherwise sort of just be emitted out of the atmosphere and reflected out. So as we see an increase in greenhouse gases, like carbon dioxide that Megan was talking about earlier, we're seeing an increase in these trapped radiation.
Oops, sorry. Great. So this greenhouse effect, as we call it, is projected to increase in over the next century. So this is a projection of air temperatures in the next century, projected by the International Panel on Climate Change, where you can see that. The warmer temperatures and these purple colors are the largest increases in temperature, whereas the more yellow tan colors are a lower increase. And right where we are, if you're in Boston, you can see we're in this little area between maybe 2.5 and 5 degrees Celsius. And we'll look at a little bit zoomed in uh, figure of this as well in a second. But to sort of tie this back to plants for all of the plant inclined people that are here and how this climate system is really related to plants. So plants influence the amount of atmospheric CO2 on a large extent. So we can see here again on this Keeling curve figure, every year there's a little uptick and a little downtick. The black line is just a yearly average. But so these red lines are an increase and decrease in CO2 that occurs every single year cyclically due to the changing of seasons. And so every year, the northern hemisphere, uh, where there's a downtick, is the northern hemisphere summer, where and spring, where plants are growing. We'll call this the growing season. So, and this isn't because plants in the northern hemisphere are better or anything than plants in the southern hemisphere. Just because there happens to be more land mass, and so the forests that are there are just overall I guess, more effective at taking carbon up out of the atmosphere. So we see this phenomenon every single year with winter creating a increase in atmospheric CO2. Um, and this is just so like one of the many reasons why Speak for the Trees and researchers care about trees is their fantastic ability to take up carbon. And specifically in the northeastern US, our forests store approximately 35% of the region's CO2 emissions. So this is like an undeniable climate solution, climate benefit that we have going on here. And understanding how climate change will affect forests' ability to store carbon is also very critical as we're thinking about the future. And so, how do trees store carbon? So, the process of carbon uptake uh, occurs through photosynthesis. So, not to throw shade at any of the algae that are producing 70+% of our oxygen, but we are tree gals here. So, we're going to be talking about trees. But in general, everything that photosynthesizes does a fantastic job at taking carbon out of the atmosphere. So we should give them due credit. But this occurs through photosynthesis, and more carbon is taken up through photosynthesis than is respired. Plants and algae also, their cells need to respire in a similar way as we do, taking in oxygen. But this, as photosynthesis is their primary driver of energy, they take in much more CO2 than they release. So this uptake comes at a cost of every time trees open their little pores on their leaves called stomae, water droplets leave, which is just what happens through photosynthesis. And this releasing of water also triggers sap flow, or the movement of water from the roots and the soil all the way up into the tree. And so this is an interesting relationship that a lot of researchers are interested in looking at, especially as weather patterns and precipitation patterns are being altered by climate change. And it's called water use efficiency. So, how effective are trees at taking up carbon while also losing water at the same time. And we'll get into why this all matters, why it's tied to climate change. But trees are large drivers of not only atmospheric CO2, but the weather even because of the process of releasing this water. But I was also just hearing something that I sort of forgot, that trees in the rainforest also produce aerosols that attach to water droplets in the atmosphere and can really just like make it rain if they are needing water. So plants are very influential on global water and carbon cycling.
Yeah, so we talked a little bit about those global projections, and we looked at CO2 on a really large scale and greenhouse gases and how they're affecting sort of the globe. And then we talked about trees and carbon storage and why we care about the northeastern United States and what trees are doing there. And so now we want to talk a little bit more about the climate in the northeastern United States. So similar to the rest of the globe, we are projected to see an increase in annual temperatures in the northeastern United States. So that little white box there is representing the northeastern US, where you can see about a 5 degrees Celsius increase in air temperatures by the end of the next century compared to earlier in the century and those temperature averages. And because air temperatures and soil temperatures are linked in the snow-free, or what we call the growing season, or when trees are primarily taking in carbon and storing carbon, we're also seeing an increase in soil temperatures in the growing season. So while we're seeing an increase in air temperatures by about 5 degrees Celsius, and people often think, okay, global warming, they understand that, we're also seeing an increase in soil temperatures, which is a little bit more conceptually difficult to understand. And winter is equally as important, if not more important, in the northeastern United States. So here we're looking at a map of the northeastern United States with that red area representing a historic area of where we would have a large and substantial snowpack from 1961 to 1990. But that white area that's colored in is the late century area, or the projected area of where that large and substantial snowpack is going to shrink to. So we're seeing a reduction in the depth and also the duration of where we have a winter snowpack in the northeastern United States. And why exactly do we care about snow? We care about snow because snow insulates soils. So a little bit counterintuitive, because when you think about snow, you think about cold and you think about winter. But if you think about like an ice shelter or an igloo, snow actually has really fantastic insulation properties because of all of the air in it. So when you have a large snowpack above the soil, it actually insulates those soils, and soil temperatures remain above 0 degrees Celsius, meaning that they don't freeze. So even in winter, you can have microbes, bacteria continuing to be active and produce heat. And all of that heat will remain trapped below ground, below the soil surface. But when you reduce that winter snowpack because of climate change, so maybe less snow falls, or that snow just melts a little bit earlier, the soils have the ability to freeze when the air temperatures get freezing. And then when air temperatures are above freezing, they also thaw. And that puts microbes, bacteria, and roots also at risk of freezing and thawing, because that heat is released into the atmosphere.
So just to summarize, the effects of the projected effects of climate change in the northeastern US are expected to increase soil temperatures throughout the growing season and decrease soil temperatures within the winters. So the work that we did at BU was largely looking at this, what the combined effects of climate change across these seasons and how these will affect water and carbon cycling, and also lots of other things. But we're mostly looking at water and carbon here that we have to share with you. So what we already know based on previous research similar to the experiment that we worked on is that warmer temperatures in the growing season increase water uptake and carbon uptake by trees overall, but that reduced snowpacks in the winter decrease water uptake and decrease carbon uptake by trees overall, while also increasing the amount of damage to roots and microbes that are really essential for making nutrients available to trees. And again, we were really interested in looking at the combined effects because these sort of phenomenon have been studied more commonly independently. But as you may all be inclined to know, ecosystems are can be synergistic or antagonistic, or lots of things can happen. So the work that we were doing was to look at how the combined effect of these warmer growing season temperatures and colder winter soil temperatures would impact all sorts of things in the forest.
So in order to examine this, we utilize the climate change across seasons experiment, which is also known as CCASE for short, which examines exactly as Megan was saying, the interactive effects of warmer soils in the snow-free growing season, and then colder soils in the winter on northern hardwood forests, the forests of New England. And the climate change across seasons experiment is located in New Hampshire in the White Mountains, if you're familiar with that area. It's right by that red star, and it's located at the Hubbard Brook Experimental Forest. So the CCASE experiment was established by Dr. Pamela Templer, who's at Boston University in the biology department. So she's the principal investigator who established this project, and she was my PhD advisor, and then also worked with Megan and advised Megan. So CCASE is an experiment that has mostly red maple trees. So when Megan and I are talking about sampling trees, we're mostly talking about red maple trees. And this is just important to note because this is the most common tree type in northern hardwood forests. And so it's really representative of what you would see in this forest, and really probably representative of whatever effects we're going to see in this forest type. And this project was established in June of 2012. So when I joined in 2016, these treatments had already been going on for quite a few years. And there are a total of 6 plots. 2 are reference, or if you're more familiar with the language, control plots. So they were just left alone, but we sort of just had this designated area where we collected all of our samples. So we knew we were going back to the exact same area every time, but we allowed snow to collect at natural levels and everything. 2 that were warmed in the growing season, and we warmed those 5 degrees Celsius above ambient temperatures based on those projected for the region. And I'll explain in a few seconds how exactly we warmed those, because it's pretty cool. And then 2 were similarly warmed in the growing season, but then we also induced these soil freeze-thaw cycles in winter. And that last treatment is getting at those synergistic effects, so seeing the combination of the soil warming and then also the soil freezing in winter. And that last treatment is really what makes this experiment so novel, because nobody before had done a treatment like this. So the way that we heat up the forest floor and then also induce these freeze-thaw cycles is with buried heating cables. So if you look at the image on the left, it's a thermal image taken during the daytime in the growing season. And anywhere where you're seeing sort of those white lines are representing a heating cable that's actually being heat. And so in the growing season, we have a program that basically calculates with these soil temperature probes the average soil temperature for the plot, and then determines whether or not the plot heating cable should be turned on or off. And you can see here that they were turned on. And then on the right, you can see a plot during snowmelt, just so that you can sort of get like the spatial extent of what the plot looks like. But essentially what we would do in the winter is we would manually remove snow, which is a fancy way of saying that we shoveled snow. And that's our way of removing that insulation based on the projected snow reduction for the end of the next century. And then we would wait for the soils to naturally freeze for 72 hours. And then we would similarly turn those heating cables on for 72 hours. And just a fun little anecdote is that the first ever soil warming experiment in the US was conducted at Harvard Forest, which is in central Mass. And when the researchers were figuring out how to do that, they weren't sure how to heat up the forest floor, because previously how it had been done, they had sort of like heating lamps, but they were like, that's more of what you would do in a grassland. How are we going to do that in a forest? And so the lead researcher was at a football game and noticed that it was snowing on the field, but that there was no snow on the field. And so he was like, how are they heating up the football field? And it turns out that they use these same types of heating cables on football fields so that the fields don't freeze over during winter games. So we use that same technology here in our experiments. But this is where it's a little bit different. So this is our shed where you can see the experimental setup. So on the left there, that's our complete control panel. So you have your fuse boxes and your relay contractors, and that's what's turning on and off almost every 5 minutes. So every 5 minutes, the system searches and basically, as I said, looks at the soil temperatures in the plots, calculates the average soil temperature, compares it to an ambient soil temperature, and then decides whether or not to turn on the cables or turn them off. And then on the right, you can see where all of our data loggers are. So that's where we store all of our soil temperature data, our sat flow data, which we'll talk about, and then a whole host of other variables like air temperature and wind direction and speed and radiation and stuff like that.
So this is a little figure that we have to share with you. The most, I guess, super sciencey we'll get. We do have extra fun data at the end for anyone who's curious, but this is just to give you an idea of what soil temperatures looked like and how we actually knew that the experiment was working. So this is from December of 2014 until almost the following year in 2015. So you can see here in this little purple box, we have the colored lines are the treatment plots, and you can see that they dip just below 0 degrees where the snow was shoveled away, and then the heating cables were turned on to get this increase, getting them back to above 0 degrees, and then leaving them alone again, and then letting them freeze, turning the heating cables on. This is just like an exemplary year where we managed to have 4 successful freeze thaw cycles happening. And then here in the growing season, you can see that the treatment plots are just perfectly elevated by 5 degrees Celsius warmed. So this is sort of our proof that everything that was intended to be going on for the most part was, but research is as it goes.
Yeah. So just to explain a little bit more my motivation. So I originally joined the Temporal Lab as a technician, and I was just really excited about climate change research. And I was also really excited about field work and lab work. And then I just became really excited about the research. And there had been 2 other graduate students who were already working in these plots and looking into different things, but then there were so many unanswered questions. And I can't stress enough how novel this experiment was, because we were looking at these combined effects, and there were just so many different directions that we could take this research. And it was also really excited when Megan joined and we could have some into the field and explore even more questions, because the more people that you have and the more energy that you have in the lab and in the field, the more you can do. So the specific research questions that the 2 of us sort of explored together were 1, how does climate change influence red maple water uptake and sap flow? And 2, are the negative effects of winter climate change offsetting the positive effects of nutrient uptake and storage, looking at carbon and nitrogen? And the way that we looked at water uptake and sap flow, first we measured sap flow and xylem. And for all of these, we're going to sort of explain our methods. This is where we get into the cool, like what exactly did we do in the field? And then we also measured soil and branch water for stable isotopes, which is a really cool novel technique that we did. And then in order to examine nutrient uptake and storage, we measured soil carbon and nitrogen content, and then leaf carbon and nitrogen content, and then also photosynthesis rates. So let's get into some methods, Megan.
So exciting. So this was totally not what I was expecting for my very first ever day of field research, but this is really what it was. So on the left here, we hired someone who I believe he was like at like SUNY. He was at SUNY USF studying forestry and related stuff, and he had been helping other researchers also shoot down leaves. So we did not shoot trees with bullets, but we did hire someone to go out and shoot trees for us. Uh, it was a shotgun with little like pellets, like BB gun pellets that were just able to nick off like the little branches that were out on the side. Because even when you have a tree climber, it's still really hard to get to the leaves that are at the ends of the branches, because obviously you can't just climb out onto a very thin branch and collect this nifty sample. So we had someone come and shoot them down from the canopy, and then my job was to run and try to catch them, or just locate them once they fell. But it was sort of easier if you had your eye on them as they were falling. And we also collected these little branch samples, which we'll explain a little bit more of. And while I was doing this,
So Megan would take those branches and then run them over to me. And we'd clip sort of the end of the branch and then quickly submerge them in water. And what you're looking at here is called a Lycor. And this is how we measured photosynthesis rates. So you'd take this chamber and you'd clamp it onto the leaf, and you'd basically create this sealed chamber where you could completely control the environment. So you were controlling the light, you were controlling the gases, and you could basically measure this leaf for as long as you want and measure for basically whatever gas the Lycor can measure for. And so we were looking at these rates of CO2 uptake based on full light levels. So something also that Megan was mentioning is we were looking at these leaves that were at the top of the canopy, like at the very, very, very top. And so in order to, if you wanted to climb the tree, you would be going to the very, very, very top. So it was easier to shoot them down. And so we were trying to represent that in this chamber by giving them like the best light conditions and the best CO2 conditions to see how much carbon dioxide they would take up in a given period of time. And then we could instantaneously measure how much carbon dioxide they would take up. We would take those exact same leaves and then bring them back to the lab. And then the next step is to
grind them up. So yeah, when I was in the lab, I was like quite literally grinding almost every day in the lab for the first summer that I was working with Jamie. And so this is what it looks like on the right if you grind up 3 leaves and put them in a cute little vial. We used liquid nitrogen to break up the leaves because I'm sure none of you have ever attempted to grind up a leaf, but it's actually very challenging, and the petioles are sort of hard because we would also dry them. So we basically had to use liquid nitrogen in order to flash freeze them and then make them easier to grind up with the mortar and pestle. And then once they were all in this fine powder, doesn't look like very much, but we actually only used like 0.05 grams to analyze for carbon and nitrogen. We had to microbalance them and then put them in a little tiny tin container that would then be put in a combustor, which would immediately combust everything and then analyze the elements that were inside of it.
And in order to measure water uptake, we used the Granier method to measure sap flow, and there are a bunch of different methods, but this is what we chose. So these 3 images are sort of looking at the sequence for how you install these sap flow sensors. So first you drill 2 holes, and they're about 10 centimeters apart. And you're drilling them right into the xylem. And then you install these probes. And the top probe, as you can see there, is a heated probe. And then the bottom probe is an unheated probe. And the unheated probe has a voltage sensor. And basically what it does is it is constantly recording the voltage. And based on that voltage or a temperature differential between the unheated and the heated probe, you can convert that into a relative rate of water uptake, or basically like the flow, how much that water is flowing. And we would install these at 3 locations around the xylem, on 3 red maple trees. So we sort of had these 3 VIP red maple trees in every plot. And those are the trees that we were shooting and collecting leaves from, collecting branches from, measuring sap flow on, all of that fun stuff. And so you can see here, we also put red solo cups on them, and then we would put these sort of like reflective shields on them too, just to protect them from any radiation and stuff like that. I also want to call your attention to right above those red solo cups, you can sort of see bands there, and those are called dendrometer bands. They are these spring-loaded bands that you install on a tree, and ideally they last the whole life of the tree. And they're spring loaded, but you have these 2 marked points on the tree, and you go to the tree however frequently as you want. You could go every day, you could go once a month, however often you want. And you measure the distance between those 2 points with a digital caliper. And as the tree grows and expands, the band, because it's spring loaded, will also expand. And because you're measuring that distance, you'll get a larger and larger distance between those 2 points. And then you can actually calculate how much that tree has grown over time. And so it's a really easy way to get a daily estimate of how much the tree has grown, or a monthly estimate, weekly estimate. And if you know the tree species, there are these really handy equations that you can scale up. This is known as the DBH, or the diameter at breast height. So if you know the diameter at that specific height, you can use these equations to calculate how much carbon that tree is storing at any given time. So just in this one picture, you're seeing how we measure both carbon storage in the tree and then also water uptake and sap flow. So that's pretty cool to see. And then also we wanted to get at sort of understanding where trees are getting their water from. And so in order to do that, we wanted to get an idea of the isotopic composition throughout the entire soil profile, because you can use water isotopes sort of as a marker to understand where trees are getting their water from. It's a little more complicated than that, but this is an example of, you have sort of your complicated methods where you go and you like set up this shed with this electrical system, but then you can also go out and like dig a hole, and that's how you collect your samples. So this is a soil pit. It takes a really long time. So it takes 2 people about an hour to dig a soil pit like this. You can see like on the edge there, the like big roots and stuff like that. And this goes all the way down to a meter. And what I like about this picture is you can see all of the horizons. You can see at the top there, like the leaf litter and organic layer. And if you look at the bottom, you'll actually see basically sand. And that's because of all of the glacial influence that there is in New England. So there's a lot of like glacial sand once you get towards the bottom, which is annoying, but also really cool. And of course, what makes field work better is to have a trusty sidekick. And so Bodhi would help me dig these holes, kind of.
And the other thing that I just wanted to mention is you can see the plot, whichever plot this is behind Jamie and Bodhi. So we wouldn't dig these holes in the plots. We actually weren't allowed to use any metal tools in the plots. So when we took soil samples, which we did take in the plots, we had to cut them with little plastic knives, which actually like broke. I think there was actually one sneaky steak knife that actually took some of the
A
salmon.
dull steak knife.
A dull steak, yeah, a very dull steak knife. But maybe it was plastic as well. But yeah, just because the heating cables, they were insulated like with rubber, but just as a safety precaution. And this is for small plots. Like this is a massive hole, which would greatly disturb the experiment and obviously the tree roots and everything. So while I was running around collecting the leaves, Jamie would be digging these holes just adjacent to where the plots were. And then once we had these lovely soil samples collected, which look super cool, we would bring them back to the lab where they would be frozen because we wanted to make sure that even, at room temperature, water can evaporate. So we wanted to make sure that all of the water was completely frozen. And then that's where the fun would happen. So once they were frozen at something around -20, definitely below -10, but the methods on that are just escaping me at the moment. And so we would keep them frozen in liquid nitrogen, which you can see are these little containers here. And I would decant them from the little jars that they originated in into these longer tubes. And we would keep them frozen in liquid nitrogen. And what you can't fully see here is like this whole tube system above, which would be hooked up to a vacuum just before we opened. There are little like locks here on these chambers. So this whole system would become a vacuum so that when we opened up these containers where the soils were, we could also open up this other side, and then we would move to boiling the soil so that the water would rise, and then we would move the liquid nitrogen to this side over here so that the water could be evaporating and then condense here on the other side. And it sounds sort of easy, but I spent many hours holding a hair dryer to the little like kinks in all of the tubes, just to make sure that 100% of the water was evaporated and then condensed, because this is really important since we were looking at isotopes. And if you're familiar with chemistry at all, we didn't want any fractionation to occur when we were doing this isotope analysis. So once the water did condense, then we could just put it in another little vial, and then the water was then sampled for isotopes of both hydrogen and oxygen. So that's just a little bit or a lot of bit of some of the research and methods that we did. And we're really happy that everyone was here to share that with us. We'd be happy to take any questions as well. And just while you're also thinking of questions, let me quickly just pull up our poll that we have for our Arbor Week events. So if you could, is the poll live and everything?
I see it.
Okay, great. Oh, there goes David. No questions,
Should
but
we
I'm sure
stay
people.
recording, or I guess for the questions? Would be nice.
Oh yeah. I think it's fine if you want to turn it off now.
I had a question. So you didn't get to use the